So my name is Kris Sankaran. I'm an assistant professor at the University of Wisconsin-Madison. I'm mainly interested in applying statistical methods to microbiome data. And one of the things I really enjoy about microbiome data is that it's kind of this interesting mix between molecular biology and ecology. So we're thinking about these tiny molecular ecosystems. All my slides are at this link. I also put it as a comment in the Google app if you want to follow some of the references. And I'm going to be talking about topic models. So topic models, old idea, one of my favorite models. Very simple, but I find it quite useful. And I'm going to be sharing some ideas in a publication that was different ideas for how to visualize topic models. Okay. But first, a little story. I don't know how many people in this room are familiar with the idea of enterotypes. This came out maybe 15 years ago, less than 15, a little under 15 years ago. And they argued that there's this kind of basic kind of clusters to which everyone belongs to, and they couldn't associate it with any kind of, it's not just the country that you're from. Right? Really, it's just they made this figure. Each point is a different person. You can see the country that they're from is in here. The country is unrelated to where they are on this map. And it seems like there are these 3 different clusters in it. Seems like a simple figure. I've never seen a simple figure lead to a New York Times article, but that's how it works. Right? And this is an interesting finding because they thought of it in kind of similar language as how people would talk about blood type. Right? So I don't know if you can read it here. Blood type meet bug type. Right? So your enterotype is like your bug type. And they're saying that these 3 fundamental bug types that everyone belongs to, maybe this has really important health implications. Okay. It was featured in Wired's Jargon Watch, right? It's not just New York Times. It was international news. This is some Dutch newspaper. I just searched for enterotype. So people were talking about this everywhere. Right? But this is maybe in the next year, there were a lot of follow-up studies. And then people realized that the story is way more complicated, right? It would be such a convenient story to just say that everyone's microbiome is one of 3 basic types, and you just need to go get a device, measure which of the 3 types you are. But really, first of all, this follow-up paper, there are people who are kind of right near the border. There's not a really obvious distinction like there was in that very first paper, especially when you start gathering more data. You start realizing there are a lot of people in between. Also, it's not like blood type, which is static, right? Your enterotype could change. This picture is tracking someone over time, and they're moving all around different kind of species compositions. Right? So I don't want to be like a statistics police, like, oh, you shouldn't use clustering. Like, that's not the message I want you out of this. The message is that by paying a little bit more attention and trying a slightly richer suite of exploratory tools, you'll be able to learn a little bit more what's actually happening in this data. Okay, so the 2 specific kinds of enriched exploratory tools that I'd like you to leave this talk with. One is that it's helpful to think in terms of mixtures. So clustering, I like to think about any kind of these modeling as some sort of data compression. Right? And how does clustering do data compression? It's taking all the samples, collapsing it down to the centroid, all these circles down to the star. An alternative way you can do compression is thinking in terms of mixtures. So instead of looking for representative center points, you look for extreme points, and then you describe points as somehow mixing between those extreme points. It's also a compression. You're representing each point by what are the coordinates with respect to these extremes. This is an old idea. This is called topic modeling. I'll review the main ideas behind topic modeling. And then the second way that you can go beyond just clustering is to think about working at multiple scales. This is kind of what our project is about. And ultimately, what we want is something like a hierarchical clustering, but instead of just clustering, you have mixed membership, or you have topic models. Right? So can you have this sort of mixture idea, but where instead of only thinking about 3 corners, you're allowed to think about 3 corners, and then 4 corners, and 5 corners. Kind of a increasing resolution topic model. Okay, so let me review topic models. So this model came out the early 2000s, I would say like between 2000 and 2003, and there were two publications, totally independently discovered the same model. One was in population genetics, the other one was in text modeling. And at first that sounds like kind of a crazy, how could the same model be discovered in two totally different literatures? But then there's a very natural analogy between these sort of population genetic, or kind of a lot of molecular biology we'll see, and text data, right? So you think of an individual as like a document, and an individual could be described as having certain kind of ancestries, like a mix of different ancestries. Right? And then what is that one kind of ancestral population? How can you understand that? You can think of it as like a distribution over alleles. And in the same way, a document, think about some newspaper article. Newspaper article you can think of as like a mixture over different topics, right? So maybe an article could be a mix of sports and business. If they're talking about some team, how people are buying teams or something like that. So it's a mix of sports and business. What does the business topic mean? It's a distribution over words, right? Certain words are more likely to appear in an article about business than an article about politics, right? So it's a very natural analogy. Actually, I wanted to put this up especially because we're thinking more and more these days about large language models in biology. And I think half of the work in trying to get that to work nicely in biology is really clarifying what are the analogies between language and biology. Okay. Before I explain the model, I want to explain the kind of outputs you get. There are two main kinds of outputs that come from LDA, or topic models. The first kind of output is this sort of diagram. This is sometimes called a structure plot. Each column is one sample. So this is from the GTEx consortium. The original data set is a bunch of samples, expression of many, many genes. The output of LDA, you have for each sample one of these bars. The colors are telling the mixtures of different topics. Right? So there are 10 different topics in this fit. One of the things that you see is that different tissue types are being represented by different kinds of topics. The other thing that I find kind of interesting is that it's not just kind of a very deep clustering or anything like that. Right? You see things like this, where there's this kind of smooth increase in the amount of blue, right? So that's saying that there's kind of some sort of gradient mixing between those two topics, right? It's mostly orange, but you have increasing amounts of blue, and it's kind of a gradual increase. It's not like a hard jump from one cluster to the next. It's you're gradually going between these two endpoints in the mixture. The second output you get from this model is like how you understand the topics themselves. So here, each row is a different topic. There were 10 topics, 10 rows. Each column is a different gene. The color is saying, and for that topic, how much are you likely to see that gene? And so like I looked up this very first gene. It has something to do with controlling the width of blood vessels. Okay? So we can go check its high value in topic 9. And then topic 9 is this gray one. So this is appearing mostly in the heart, which makes sense. And if you look more carefully, it's not just 9 that's expressing this very highly. Right? Topic 2 and topic 1 also seem to have high values. So whenever you have high membership of topic 1 or 2, you also tend to have a lot of this one gene. You go back to 1 and 2. 1 and 2 are this blue and kind of yellowish. Right? And so that's happening in blood vessel, it's happening in blood, lipid, and muscle. And so this is kind of all consistent. And this is basically how you interpret these sort of topic models. A second example, so this is a project I worked with people who were interested in what happens to the microbiome after people take antibiotics. So the analogy is like a wildfire sweeps through an ecosystem, a lot of the initial animals not there. How does succession work? How does colonization work? So I always love these sort of complicated data visualizations, but sometimes it's just not useful. Right? If you just try to highlight everything, it's hard to see what's going on over time. Right? This is a situation where a topic model might be helpful in getting a better summary. Okay. So this is what happens when you apply it. Now each column is one time point for one person. I've divided up according to there was one treatment of antibiotics, they took a break, and then there was a second treatment of antibiotics. The main patterns you see, right, there's kind of this blue or there's like a topic that's blooming during the antibiotic treatment. Pink basically disappears. It only really slightly appears after the second antibiotic. There are these kind of different topics that are emerging during the interim that are different from the initial topic. And maybe the difference between blue and gold is is already present before, and then green seemed to really only emerge during the interim. So this is exactly analogous to the structure plot I showed for the gene expression data. And then the way you interpret the output. So here's a different way of interpreting the outputs. What I've done is I've taken species where they have very high kind of topic score or like topic probability for one of the topics and low values for all the other topics. So these are somehow like the purer taxa. And it's consistent with what we just saw, right? So these are specific species that are increasing during the antibiotic phase. There are some that are decreasing and rebounding. There are some that are decreasing and never really coming back. Okay, so that's output from LDA, or topic models. Formally, how does this work? This is the entire model. Think of xi as like all the gene expression. So think back to that GTEx data set. Xi would be like, what is the expression for every single gene? Ni is like the number of reads for that sample. And where does the multinomial distribution come up? Multinomial is what happens when you flip a die, right? So 6-sided die, flip it, the probabilities of each of the face, that's the multinomial distribution, right? Or the number of outcomes you see for all the different faces of the die when you flip it many times. So how's it coming up here is you think of, for every single read, you flip a die, and then you see which of the genes does it align to. Right? And then you're just counting the alignment across all the different genes. The key point is the probability vector, so the probability across all those different genes, is somehow being shared across all the different samples. And the way that it's being shared is through this matrix B. This is a gene by K matrix. Right? This is K columns. The K are the different topics. What it's saying is like you have these, think of K as like 10. You have 10 of these kind of essential die. And those are die that are giving you the probability across all the different genes. And for any one sample, you're kind of defining a new die that's a mix of all those 10 essential die. Right? So if you're kind of very close to one corner, your mix is, you're basically using one of the die and kind of ignoring all the other die, and that's how you draw gene expression values. The gamma I is telling you what is your mix across those 10 die. No, you have to estimate them. Yeah. And that's why actually here we're putting some priors on what the topic is. Right, so number of K you have to choose. And that's actually going to be what the whole second part's about. Yeah. Other questions?
So we are actually clustering the topics.
What do you mean by clustering topics?
We estimate the topics, so it is kind of a clustering.
It's kind of like you can think of it almost like a soft clustering, except you're not just assigning to one cluster. If the gammas were all zeros and ones, then it would be assigning to one cluster. But since gamma can be a mixture, right, it's like in all these pictures, each sample is a mix of different topics. Right? So it's a little bit richer than just clustering. That's kind of what we're trying to get beyond interotypes. Okay. Other questions? Okay. And just to give some more geometric intuition about those formulas, there are two kinds of simplices that you have to have in your mind. One is the simplex for understanding the gamma i's. Each corner is one topic, and this is exactly the same picture that I showed for understanding clustering, right? So instead of clustering where you represent by one point, you represent each sample by mixing across different underlying topics. The second picture you have in mind, and I haven't drawn this one yet, is the simplex that represents the topics. So you think about one topic as kind of mixture over genes, right? So if I had you can only draw 3 corners and have it make sense, but really it's like the number of genes, dimensional simplex, and if you're very close to one of the corners, that would say that you have a lot of one gene and very low for the rest, but really you're usually somewhere in the middle. Okay? So those are going to be the columns of B. This is going to be the gammas. Okay, so that's topic modeling. So now I want to talk about alignment, and this is what our project was about. So this already came up, the question of K. Right? And this is the main parameter for working with these topic models. This came out, even in the original, this is the Pritchard, Stevens, Donnelly paper. This was already kind of understood to be some issue, right? Like, how do you actually choose K? It turns out statistically it's difficult to estimate. Right? The sort of marginal likelihoods are very difficult to compute. There are also biological reasons to be a little bit concerned. In practice, if you're just using this routinely, it's common to just fit across many different K, trying to make sure that whatever you're interpreting is sort of stable across many different K. Okay. Yeah, and actually kind of the starting point for this project was I was in some lab meeting discussing with a collaborator who was applying some topic model. They had spent a lot of time trying to understand exactly what is the right K. They had been trying all these different tools for it. And then kind of my thought was like, do we really even need to choose K? Is there no way you can actually try to understand all the different K and understand, you don't have to choose, but just try to be reasonable? So like one example is like marginal likelihood. Right. So yeah. But there are other proposals out there. When K is 1, it's like representing everybody by one topic, right? It's just like you take the average probability across all samples. It's like the average expression value. Okay, so this is the main idea for how we do this alignment across K. How do you read this diagram? Each column is a different model. Each rectangle is a different topic. The heights of the rectangles are the total amount of membership that across the entire sample that go to that topic. So like in that picture with the GTEx and you had the gray for the heart, it's like the total area of the gray, that would be the height of this that corresponding topic. If you have like a topic with very little color, it would be a narrower topic. And then alignment is this, we draw connections between the topics if they're very similar in some sense. Yeah. And it's not necessarily a tree. You can have some merge back and forth in the tree. But you get this sort of tree-like structure. The trouble is going to be, how do you actually define these sort of edges? And what does it mean? Can we give any sort of clear interpretation about it? Okay, just establishing some notation. We think about this sort of like a graph. I mean, it is a graph, right? I'm going to be using V to index each of the topics. I'm going to be using E to index the connections between the topics. We're only going to be considering edges that are between models with K and K plus 1. Right, so we're not going to be drawing edges between very far away topics, even though technically you could. The information associated with each node, we have, again, it's the betas and the gammas. Different dimensions. Beta is across the genes, gamma is across the samples. Okay. And if you start thinking about it as a graph, this actually simplifies a lot of the notation, right? Instead of having to index K and M, so K and M were all the different models, the different Ks within each model, now we just need to index it by the node. So when I write later on you see V, I'm just referring to a topic within some of the models. You don't have to actually memorize this. Only the kind of important ones are going to be gamma V, that's like the membership across all samples for that one topic, and beta V, that's the distribution over features, genes, bacteria, metabolites, whatever you want. So how do we define the edges between the topics in this diagram? We have two ways. The first is kind of the most simple one. You just take the inner product, it's like the cosine. And the intuition there is, if across the n different samples, this one topic from some model has a very similar distribution across the samples as another topic, then you kind of think of them as being similar topics. Right? So like if they have high weight for all the heart and blood vessel samples and low weight for all the others, then you think those two topics are similar. That's what this metric captures. Yeah, it helps. Everything is positive. So that's why that metric still makes sense. Okay, and a second way you can think about it is using optimal transport. So here, this triangle, this is like the second triangle that I showed for defining the topics. Each of these columns is a topic. The colors are representing what the K was for the original topic model. So there's a topic model with 2 topics, one with 3 topics. The heights are the amount of membership across all the samples. And you need to learn, kind of for any one topic, so say this one purple topic, you need to learn how to transport the mass to topics from the other model. Right? And you want to do this in some sort of efficient way. And that's going to, those kind of transport plan are going to turn into the edges. Okay. So formally, this is how you can write it. C is a cost for transporting from one topic to the next topic in another matrix. W, this is exactly the pi from Ben's talk last week. It's a transport plan. These are constraints on the total mass within each topic. The intuition is, the analogy I really like is something like kind of one set of topics is like mines, another set is like factories. You're trying to transport material from the mines to the factories. Each mine can only produce so much material. They have different kind of productive capacities. Each factory has a different kind of demand, and different kind of demands across each. The factories and the mines are located somewhere in space, and you want to figure out how to transport material from the mines to the factories in a way that's very efficient. You don't want trucks going a very long distance if they don't need to, and they can still satisfy these constraints. Okay. So that's essentially what this formula or this problem tries to solve. Just trying to make it as explicit as possible in this example with 2 topics and 3 topics in the 2 models. These are just 2 matrices with 6 entries each. Right? Your costs, and then you try to choose your W so that you avoid the entries with large cost while still satisfying these kind of sum over the rows, sum over the columns constraints. Okay. Something that you can do once you have these diagrams is you can start coming up with different kinds of diagnostics that are not just one model at a time. The kind of traditional diagnostics topic models, you just fix one model and then you compute like a marginal likelihood or some goodness of fit for that one model. But now that we have this diagram, we can start coming up with diagnostics based on the pattern across the ensemble of models. So what are the kinds of diagnostics you can come up with? One is you can count the total number of paths that appear in any model, or in the ensemble up to a certain point. So it's like when you go back, you notice they're all colored by, like there's a shared color across all the topics. The total number of unique colors up to a point, like that's one thing that you can measure. So that's, when I call it paths, I mean kind of these shared colors across the diagram. Coherence, I don't know. I'm not sure where coherence and refinement, the names came from, but the idea for each coherence is we're saying for any topic, you look at the similarity to other topics on the same path. If it's very similar to other topics along the same path, then it's somehow more stable. We think that that's more likely to be a real topic. Versus if it's not very similar to any other topic along the same path, then it's maybe like a spurious or like a transient kind of topic. And then the last idea was refinement. So if you have something branching off like this, where every topic has only one parent, that's somehow a cleaner, maybe more real structure. You're not kind of introducing random topics unnecessarily. Versus if you're in this situation where they have many different parents for each topic, then it's like, it's just some cloud of points and you're kind of randomly moving topics around. Right? Here you expect there's a real hierarchical structure, here this is just noise. I have formulas for how we compute this, but that's not really the point. It's just capturing this intuition. Okay, are there any questions about the main idea before I go into some examples? Yeah?
So why is the top one as the higher, I mean for B, why is the top one as the higher coherence than the top?
Okay.
Can you repeat the question?
Oh yeah, so why does this have higher coherence than this? I'm trying to, like, you think of this as one path, and then think of this as another path. This has a large edge to here. And then this, I think it's just that the edges are going to be smaller to any of the nearby, or any of the others on that path. I drew this one because this sort of edge never actually appears in the diagram I made, right? This is across many k. I'm actually computing extra edges. And you want the edges to be very wide, even when you jump more than 1k away. I think that's the main intuition.
Isn't it because they are between two tall rectangles, the top one, that's why they're here?
Yeah.
One is between a tall
Like, yeah. Like what probably should have drawn is like exact same height for the endpoints. And then like same height for the endpoints, but then have full edges for the top one and then narrow edges to those endpoints for the bottom one. Does that make more sense? So it's like even along the same path, if it's not neighboring, you want it to have large alignment weight. Yeah.
So in terms of the term high coherence versus low coherence, is it similar to like highly connected components versus like weakly connected components? Because I'm trying to understand this in terms of like graph network analysis. Because it seems like the concept is
it's similar. Yeah. It's like you could have very high flow across that path. Would be kind of like relate it back to actual graph theory, right? But I don't know if it actually matches exactly with any graph theoretical idea.
And in terms of the alignment, why? I mean, your method is one method, and we're trying to experiment with like fuzzy logic matching. So have you considered that method?
Oh. I haven't worked with fuzzy logic before, but it has a kind of similar similar issue where it's like it's almost like what you would want to do with hierarchical clustering, but I want everything to be kind of mixtures, right?
I mean, in our knowledge graph, it's pretty highly mixtured, and we're trying to kind of look at across different knowledge graphs and find either strong, like high, refined or coherent patterns, or understand certain topics that are weakly connected components, like why those are weak and how that influences, like in terms of like machine learning results. So, but when it comes to aligning different knowledge graphs, it's such a headache because the edges are not that straightforward alignment. Nodes, yes, somewhat, but in terms of edges, there's a lot more questions how we can align edges compared to like nodes. Nodes are pretty straightforward as long as the nomenclatures are pretty much the same.
Yeah, no, I mean, I think this is getting into more complicated.
Yeah.
But this is, I mean, that's definitely starts getting more interesting. Like, instead of these kind of fixed K topic models, could you think of alignment productively in like a less structured, more generic way? We've actually been thinking about similar ideas for data integration, right? So like, instead of just, can you align different modalities? So this is a similar kind of generalization. That'd be interesting. Okay. Other questions? Okay. So now I want to share a few examples. Okay, I have a question for you. So I generated data. Here's from an actual topic model, 250 samples, 1,000 features. What is K? Anyone want to guess? Okay, why? It is 5, but I don't know. Yeah, do you want to explain why?
It's like the last place before things start to like trickle off.
Yes. I mean, yeah, that's basically, I mean, all this effort of coming up with names for things is we're trying to capture this sort of intuition, right? Yeah, it's like up till 5, you define a new kind of large topic, and then afterward, it seems to always introduce just these small topics, right? The other way you could have told is that there are 5 main colors in this path, right? There's a 6th smaller color that appears here, but that's the only place that it appears. Otherwise, it really partitions into 5 colors. Okay, this is good. And you don't just have to guess from the picture. These metrics that I just introduced, they also sort of capture that K equals 5 is the important number of topics, right? So if you look at the number of paths, the number of colors in the diagram, as you increase K, up until 5, very clear kind of monotone, and then it just stabilizes. Transport seems to be a little bit over-optimistic in defining topics. But in the previous diagram, we already saw that the ones that it tends to introduce are very small topics, so you can still visually tell, even though this metric doesn't quite get it. These are a little bit more complicated. I've repeated this kind of simulation 200 times. Each point is one topic from one of the simulations. The color is the similarity between the estimated topics and any of the true topics. So you never actually know the color in real data analysis, right? Because you don't know the true topics. But what's interesting is that we are seeing kind of this coherence and refinement scores. They do pick up on the difference between the topics which are really related to a real one and the ones which are just noise. Right. Yeah, and it's kind of increasing to K, and then there's a drop-off. And that happens in both. Okay, this is a big diagram, but I have 2 main points to share. One is that the quality of these sort of diagnostic measures depends on the sample size. Right? If you have a very small sample size, these aren't really that sensitive at detecting the true K. Larger sample size, we do start seeing like some more concentration around the actual K. We also see this issue that we saw in the previous one about the transport sometimes having too many topics, while the simple kind of just inner product one really does stabilize at the right K. Okay. So one of our goals here was also to think about, are there kinds of departures from the true topic model that we can be sensitive to? Right? If the model is misspecified, can we say something productive about in what way it might be misspecified? So here's one simple way to misspecify a topic model. Beta gamma I, that's exactly the formula from the original topic model. So when alpha is 1, this is just an ordinary topic model. But I'm also adding in this sort of thing of like contamination. For the contamination, I'm just drawing these new i's from across the simplex. So it's kind of information that's not shared across any of the samples. There's some noise I'm adding in. So I can simulate data from this model. I can fit this sort of alignment, and we can look at the diagram. So here is the just kind of random Dirichlet noise. Here is the true K equals 5 topic model. And then we can mix between the two. Right. And the diagram does seem to kind of transition more smoothly between them. And again, in the metrics. Right. So this is kind of closer to the real topic model. It's 0.8. And then down here, this is like you're only having noise. There's no low rank structure by the bottom. And you can, it's kind of nice that this pattern of this drop off basically disappears by the time that you don't have any low rank structure. So this diagnostic is doing sort of what we want to do, and it helps us get some sort of reference point. Okay. Second simulation, this is a problem that we've read about in some applications that are about something they call strain switching. So the idea of strain switching is you have two communities that are basically the same, except on a subset, you measure one group or the other. Right, so here is, these species are the same, but in this kind of version you have just these three present, in this version you have just these three present. Right. So in order to try to understand this sort of phenomenon, one idea is you need several resolutions. Right? You need one resolution just to realize that there are these two versions. There's this kind of, that all of these are the same. And you need a second, finer resolution to realize that there are some samples which are closer to this one, and there are other samples that are closer to this one. Okay. So we defined a simulation mechanism that tries to capture this intuition. We took a bunch of these topics, defined the k, I think it's, again, k equals 5. Maybe we'll see in the result. We have some set of topics that are the true topics, and then for each one of them, we perturb them on a subset. So it's just like this. I defined the blue, and then I took kind of subset of species, and I just switch between groups. Okay. This is the output diagram. And then the point is, yeah, so this is k equals 5 real topics, and then for two of them, I defined perturbed versions. Right, so when k equals 5, it does get these kind of larger level topics without noticing any of the switching phenomenon. And when you get to k equals 7, it's disambiguating the main topic into smaller versions that are almost the same, but on a subset of species, there's some difference. Right, I think like out here, there's some difference compared to here. Okay, that's that simulation. Okay. And this is again you can look at the relationship between, like the similarity between the estimated and the true topics. When k equals 5, it merges those two topics that are almost the same, except for the subset. It's different. K equals 7, you split that out. Right. They're still pretty similar to the, like the two perturbed versions are quite similar to one another, which is why it's not totally red here. But you are able to split those two topics out. So this is another example where doing something multi-scale, thinking hierarchically can help. Okay. So now I want to give a real data analysis example. This is kind of where the whole project started from. So this was a collaboration with Laura Symul at UC Louvain, and she's interested in a lot of the vaginal microbiome kind of research area. So let me give a brief introduction to what that is before explaining the data that we have. So historically, kind of one of the first big publications in this field was defining something called community state types. You can think of it almost like enterotypes, which are the vaginal microbiome. Each column is a sample, each row is a species. The big difference, this field compared to enterotypes, is that for 4 of the groups, it's just one species that's dominant. Right, so there's Lactobacillus inners, Lactobacillus crispatus. For a lot of people, you only have one of these species. Right? And then there was this last CST, which is kind of the everything else CST. And there are people, it seems like they have many different kind of bacteria. There's no obvious dominant player here. So now the thinking is, in the same way that we said that mixtures can help us get a little bit beyond clusters, is there any kind of additional structure in that kind of catch-all CST? And the thing that really helped Laura in her study is that she had many more samples than that initial publication. So she had almost 2,200 samples from 135 people. It was a longitudinal sampling. First things that you get out, the blue and the green, these are these 4 lactobacillus CSTs. These are the ones that are dominated by just one type of lactobacillus bacteria. Right, so that recovers some known knowledge. Right? And then now we are kind of interested in these blue and the orange and the red ones. That's dividing out this kind of catch-all CST from before. Okay, so we can find these sort of coherence scores overlaid onto the topics. Yeah, so this is going to help in actually interpreting which ones are worth studying more. I think the thing that was the most interesting to me about this plot is that it's not really just a function of k. Right? In all the other simulation outputs, you notice it kind of increasing quality up to the right k, and then it decreases right after. Here, it really depends on which topic you're on. Right? So there are these lactobacillus topics where it's very clearly defined for any k, right? And then you have these high scores all along the path. There are others where it kind of increases. There are others where it kind of has a peak and then it decreases again, right? So here it kind of increases out here and then it starts decreasing again. Right? So I think it could be helpful for trying to pick topics at different levels that you think are the high quality topics, rather than just picking one model. Okay, so that one was, the first diagram I showed you was k up to 9. This one was k up to 15. And here I'm just computing some summaries up to k to 25. There's not as clear drop-off like in the simulated data. These are always more complicated than simulated data. Right? But the main points we observe, there's kind of a drop-off from 5 to 6 for this cluster of topics. Right? And then kind of again, like 8 to 9, 11 to 12. Some of these lower group of topics seem to drop off. So those are where we concentrated our attention mainly. Right? Whenever the score seems to drop off, right before there, that's the level of topic that you use. Okay. And this is exactly like that original GTEx plot where I have the distribution over different genes. Now it's just across the species, and it helps relate the different models. So this is the k equals 5 model, this is the k equals 9 model. These are kind of the levels where we thought there was some sort of reasonable structure. And then you can start breaking it apart. So right, like what had been this topic 4, C0, becomes C0, but also C1. Right, so there's a little bit of, there's some samples that are a little bit closer to 1 than the other. Right? If you only had 5 topics, you'd have to kind of group them all together according to this topic. But if you're allowed a little bit more granularity, you can split it into those two. Okay. And you could also make the time series, because these were longitudinal data. Okay. So for the first 2 weeks, then this green topic, what's kind of interesting is that they mix these 2 topics in the next few weeks. Right? So this is kind of, again, why it's helpful to think in terms of mixtures rather than just clusters. Right? So it's a combination of those 2. And there's a small phase corresponds to bleeding, where this orange topic increases and the blue topic decreases. So this is, you think of it like there's some continuous going between the corners, and then you kind of move a little bit closer to one corner, a little bit further away, depending on some external factors. Okay, so that was the data analysis. I'm about to wrap up. I want to compare it to some ideas that you might have already heard about for evaluating topic models or thinking about hierarchy in topic models. Okay. So what's similar, what's different? Okay, so the first thing you might think about is this: you can compute test set perplexity, or this is like the marginal likelihood, right? What's similar, what's different? So they're both for choosing k. The main difference is, one is it doesn't give topic level quality, it only gives kind of model level quality. And then, I mean, one advantage is that test set perplexity is really explicitly about looking at a holdout set. While everything I've talked about here is only on a kind of the training set, or just the data. Right? There's no sense of like generalization. Right? Okay. Also, superficially, what I've drawn looks a little bit like hierarchical topic models. And in our paper, we have a small section where we apply hierarchical topic models to the same data set. But in practice, they end up being quite different. And the main way that they're different is in hierarchical topic models, the hierarchy is as you sample each word. Right? If you're sampling a word from the document, the word is assigned a path along some tree of topics. Right? So it's a very different generative mechanism. Right? Versus all we're doing is we estimate the models first, and then we, post-estimation, build a tree structure to help understand the result. Right? A practical advantage is that this ends up being much more memory intensive. So we couldn't even run it on the entire data set, like a relatively small data set from the data analysis. Okay. And I'm happy to explain a little bit more some of those differences. Okay, everything's in the software package. Just want to include a small example. This is the actual data set that I just showed. Only a couple lines. You can fit the models and get the topics. Okay, so that's the takeaway. I think it's a relatively simple idea. This is actually this paper. I was very excited when I was writing it. Afterward, I thought, this is too simple. Like, this is no one, why would anyone care? But now, again, I kind of, it's one of the ideas I've had that's been actually practically useful in a lot of my follow-up. And that's all. So thank you to my collaborators, my group, funding. Happy for any questions.